Hello students, how are you all? So I hope your score must be going good. So how is the score? Score is the exam which I hope that and I expect that all of you must have enrolled by now, and this is an exam which will help you to make yourself ahead from others. It will help you to evaluate yourself, and as I promised you earlier that for score I will train you for the botany subjects, and similarly in other subjects also the sessions are going on. If you are regular with these sessions, then definitely you are getting to have a very good performance in the score. Not only that, it will also create a very good foundation for NEET exam. So here score, if I say, it is not only a way to get scholarship, but it will also be an opportunity to get a lot of cash rewards. So what are you waiting for? If you have not registered, register now and get an opportunity to evaluate and participate with the best mind of India, where you will have a national level exam and ultimately your academic talent will be evaluated and rewarded. So in this session, we will be talking about some important questions in botany, which will be kind of a high yielding question for a score exam. These questions will not only help you to do good in score, but also build some basic concepts. So let us start without wasting time. Some of the previous year questions, here question will be from score, question will be from NEET, and some of the very important questions from the botany chapters which are part of the score syllabus. You know, cell cycle, living world, biological classification and morphology. These are the chapter syllabus for the score. So questions from all these chapters are there. So let's start. So the first question from living world. So here the biological organization start with. So I guess all of you can answer this question. It's a very basic question. Biological organization starts with, biological organization, it starts with, just a second. Yeah. So biological organization, we know that it starts with cell. We know that cell is the lowest level, or it is the starting point of biological organization. Cell forms tissue, tissue forms organ, organ forms organ system, and this organ system form organism. Now this is for animals. This is for animals. For plants it will change. How it change? So this will be new. Just keep it in mind if you have to go for the plants. So in plants it will be cell. Cell will form tissue. Tissues will come together and form tissue system. Tissue system will come together to form organ. And organ will come together to form organism. So what you are seeing the difference, that in case of plants, you are not seeing organ system. Organ system is only present in animals, whereas tissue system is only present in plants. Remember these are the level of organization in animals and level of organization in plants. So from here, this is the question. In both case, it starts with cellular level, and anyway we call cell as the fundamental unit of life. Second question, the lowest category of plant that is characterized on the basis of both vegetative and reproductive features. Living world. You may recall in our last session where we have covered 3 chapters. There I have told you that there are 7 categories, or we call it taxonomical categories, right? So these 7 categories are basically the taxonomical categories. What are the taxonomical categories? These 7s are, I have told you the trick to remember it: king, Philip, cut, open, 5, green, snake. K for kingdom, then phylum, then class, order, family, genus, and finally species. So here we say this one as the lowest level of organization, and this one is the highest level of organization, right? So on that basis, the lowest category is the species. Clear? So species is the lowest category. Remember, there are 7 taxonomical categories arranged in this clear order. Remember the order like this: king, Philip, cut, open, 5, green, snakes. In this order you have to remember, shortcut to remember it. Then during gram staining, which is incorrect. Now gram staining, what are gram staining? Gram staining, if I say gram staining is a technique developed by, who developed gram staining? It was Christian Gram. He developed the technique of gram staining. What he did? He stained bacteria with which dye? With the crystal violet. And when he washed it, he find some bacteria which retain the stain are called gram positive, and the one which lose the stain are called gram negative, right? So the one which retain the stain, you just put a t-shirt in a yellow color, you take it out, it is yellow. Now you wash it again. If it lose the color, means it lose the stain. It again become white, means it is gram negative, didn't retain the stain. But if it remain yellow even after washing, it means it retained the stain. So the one which retained the stain is gram positive. So here if we see options, during gram staining, which is incorrect, all bacteria take crystal violet stain. Has all bacteria taken crystal violet stain? No, the gram negative lose the stain, whereas gram positive retain the stain. So that is the difference. So all bacteria take crystal. This is wrong. Gram positive bacteria take crystal violet stain. Yes. Gram negative bacteria take? No, negative do not take. Gram negative bacteria lose stain after alcohol. Okay. Yes. So here basically when we say lose the stain, so here we may call this point, differently, and let's say if we consider these two. So remember what I said, when we put the t-shirt in the yellow color, all will get the stain, but what will happen? Once we wash it, after washing, they will lose the stain, right? So both will take the stain, but gram negative will lose the stain after the, so this is also correct, this is also correct, and this is also correct, right? What is wrong? All bacteria take the crystal violet stain. Is it clear? So once again, very important experiment. I am again repeating. Christian Gram use crystal violet to stain bacteria. So both of them take the stain, but after washing, one lose the stain, one retain the stain, right? So that is why these two statements are also correct, and this is also. So first one is wrong. Next, The non-membrane bound. Now this is the question from chapter cell, right? So non-membrane bound organelle found exclusively in animal cell. First of all, two points are there. Non-membrane bound organelle and exclusively in animal cell. These are two important points. So here, if I say non-membrane bound organelle, so first of all, cell organelle. Cell organelles are of two types. First of all, it can be with membrane or without membrane. With membrane and without membrane. Without membrane are ribosomes. Ribosomes are cell organelle without membrane. What else? If I say inclusion bodies, then centrosome, or you can say centriole, then even the microtubules or the cytoskeleton we call it. These are non-membrane bound cell organelle, whereas membrane bound cell organelle is further divided into two types. One which has single membrane, another which is double membrane. The one which is double membrane, example, double membrane example are mitochondria and chloroplast, or you can say plastids, right? Plastids which include chloroplast, they are double membranous. Another one is the nucleus. Double membranous cell organelle. Single membrane bound cell organelle, ER, Golgi, lysosome, vacuoles, etc. These are single membrane bound cell organelle. Is it clear or not? So with membrane can be single membrane bound, double membrane bound, or without membrane. Is this point clear? So this is very, very important point from where questions can be asked. So remember, now on that basis, if I say here non-membrane bound cell organelle found exclusively in animal cell. In animal cell, so animal cell have this centrosome or centriole. So here what will be the answer? Centriole will be the answer. Right, very good. Okay, so Chutki has answered. Okay. Correct answer. Option 4 is correct answer. Next one. Which of the following is the largest constituent of membrane of erythrocyte in human being and is also responsible for performing most of the function of the membrane? Again, very interesting question. See, this is related to the cell membrane. See, whenever we study cell membrane, which cell we use to study cell membrane? It is the RBC. RBC is used to study the membrane. Why? Because it do not have nucleus and also it do not have maximum cell organelle. So what is predominantly present? Membrane is present predominantly. So RBC is used. Now in RBC what we study? That main composition is lipid and protein. What is the percentage of lipid? 40%. What is the percentage of protein? 52%. So erythrocyte, which is the most, 52%. So it is the most important component. So it will be protein. Protein is the right answer here. Understood? Although remember, not in all membrane protein will be maximum. It is the erythrocyte where it is maximum. In different membrane, the composition will keep on changing. Next one. Cell is the fundamental structural and functional unit of all living organisms. See, it is the definition: fundamental, structural and functional unit. NCERT line. Pure NCERT line this is. Yes, it is correct. Anything less than a complete structure of cell does not ensure independent living. Now this is also NCERT line, right? If you go and see the prokaryotic section, there in the basics of cell, what is cell paragraph, there this line is given: anything less than the complete structure of cell does not ensure independent living. Then here question comes, what is a complete structure of cell? Means what should be present so that we can call it a cell. So to be called as a cell, there are three important components. If these three are present, then we will call it a cell. If any one or more is not present, then it is not a cell. And what are these? These are, there has to be a cell boundary, right? Then it should have a genetic material. And lastly, it should be having its own metabolism. If these three are there, then it is fine. We will call it as a cell. Otherwise, we will not call. For example, virus we don't call as a cell. Why? Virus do not have its own metabolism. Virus has cell boundary, right? Boundary is there, although not cell membrane, but yes, boundary is there. Genetic material is also there, RNA or DNA, but do not have its own metabolism. So anyways, coming to this question, the complete structure of cell does not ensure independent living, and this is the reason why it is called as the fundamental structural and functional unit of life, right? So these points. Now, these are also the metabolic reaction can only occur inside a living cell. Is it true? Metabolic reaction can also take place outside the living cell. Means in vitro it is also possible. Wrong. Nucleic acid present in the cell ensure living state? No. Nucleic acid doesn't ensure living state. Living state is ensured by this. The membrane bound organelle cause differentiation for proper functioning. Again, not relevant statement. So first one is correct. Then in 70S ribosome, S stand for sedimentation coefficient, correct? S stand for sedimentation coefficient. Only prokaryotes have 70S ribosome. Now prokaryotes have 70S ribosome, but if I say only prokaryote have 70S ribosome, that is not correct, because 70S ribosome is also present in mitochondria as well as chloroplast, right? So what should be the answer here? See, what should be the answer? Statement 1, 2, both. So here in 70S ribosome, S stand for sedimentation coefficient, correct? Only word is wrong here, because eukaryote have both 80S and 70S ribosome. So only is not the correct word. Next, state the following statement as true or false. So this is a true or false type of statement. Important, such statements are there in your exam. So first, integral protein are partially or totally buried in the membrane. True, very much true. NCERT. Human RBC membrane has approximately 52% protein. Again true. Protein move laterally within overall bilayer of the membrane. This is also true. So all three statements are true here. Polar molecule cannot pass through lipid bilayer of plasma membrane by self. Now polar molecule means molecule which has charge, right? Polar molecule, molecule which has charge. Now if charged molecule has to pass through the membrane, then membrane cannot allow. Why? Because of this. See, membrane is bilayered structure, right? When it is bilayered, it has head. It has head. This head is hydrophilic. And the tail is hydrophobic. Now, see, whenever a charged molecule, whether positive or negative, has to pass through it, it has to pass through the hydrophobic layer. That's why any charged molecule cannot cross through the plasma membrane by itself. Then what? How can it pass? It needs a specific protein to pass through it. So let's see the statement. Polar molecule cannot pass through lipid bilayer. Yes, it cannot pass, right, by itself. Polar molecule require carrier protein to facilitate their transport across the membrane. This is also correct. Yes, right, a carrier protein is required. So both statements are correct. Then coming to the next one, plasmid are extra chromosomal DNA. Let's see. If this is the, now let's say this is the main chromosome, right? So this DNA of the main chromosome, sometimes we call it chromosomal DNA, right? And the DNA which is present, or the DNA which is present outside, this we called as plasmid or extra chromosomal DNA, also called as extra genomic DNA. This we called it as genomic or chromosomal DNA. Genomic or chromosomal DNA. So is it clear? So plasmid are extra chromosomal DNA. Correct. Plasmid are found in bacteria and useful in genetic engineering. Yes, this is also correct. But is the explanation correct? So whenever you have to solve assertion reason, connect both of them with because. So let's read it with linking with because. If we say plasmid are extra chromosomal DNA because plasmid are found in bacteria and useful in genetic engineering. No relation, right? Not much of a relation here. That is why not a correct explanation. Now this we have just done that 70A. So this point is already done. Now let's match this one. Amyloplast. See, these are different types of plastids. See, plastids are of three types. It is chromoplast, chloroplast, and leucoplast. This leucoplast is divided into three types. Now, leucoplast is used for storage of food. Now, when it store oil, it is called elioplast. When it store oil. When it store carbohydrate, then it is called amyloplast. Carbohydrate. And when it store protein, it is called eleuroplast. So all the types of All the types of plastid is clear here. So now on that basis, if we solve this question, amyloplast. Amylo means, here we can say carbohydrate. Carbohydrate it will store. Elioplast will store oil or fat. Chloroplast. Chloroplast, it is green in color. Chloroplast is green in color because of chlorophyll, and chromoplast is also colorful but color other than green, and leucoplast is colorless. Colorless. So now let's see here, let's match it. So store oil or fat, correct. Amyloplast. So here you have to mark the mismatched pair. Okay. So amyloplast store protein and granule. Amyloplast store carbohydrate. So this matching is wrong. This matching is wrong. This matching is correct. Chloroplast contain chlorophyll pigment. This matching is correct. Chromoplast contain colored pigment other than chlorophyll. So this matching is correct. This matching is correct. This one is wrong. So this information is very, very important. Then coming to the next. Endoplasmic reticulum, Golgi, lysosome, vacuole are grouped together as endomembrane system. Yes, endomembrane system, they are grouped together. There are 4 components of endomembrane system: ER, Golgi, lysosome and vacuole. So correct. The function of endoplasmic reticulum, Golgi, lysosome and peroxisome are coordinated. Yes, they are coordinated. But ER, Golgi, lysosome, what they have done? They have tricked you with peroxisome. Right? Peroxisome, is it a part of endomembrane system? No, peroxisome is not a part of endomembrane system. So reason is wrong. Assertion is true. Reason is false. Next, consider the following statement. Nucleoplasm contain nucleolus and chromatin. Nucleoplasm contain nucleolus and chromatin. So, inside the nuclear membrane, let's consider this is the nuclear membrane. What is present? Chromatin and the dense structure of nucleolus. So, this is correct. Nucleolus is separated from the rest of the nucleoplasm by membrane. Is nucleolus surrounded by a membrane? No, nucleolus is continuous with the nucleoplasm. So this statement is wrong. Nucleolus is produced from and associated with a specified nucleolar organizer region. Correct. NCERT line. And the nucleolus is the site of rRNA synthesis. Again correct. So 1, 3 and 4 are correct. Second is wrong. Then now we are moving to the cell division part. So here meiosis has evolutionary significance because it results in, what does meiosis result in? Meiosis result in genetically similar daughters, right? So genetically similar daughters, it is the characteristic of mitosis, not meiosis. 4 daughter cell, yeah, 4 daughter cell is true for meiosis, but is it the reason for evolutionary significance? No. Eggs and sperms, they are formed, but still main reason is the recombination. Recombination will lead to variation, and variation lead to the evolution. This you will find in the paragraph which mentions significance of meiosis. There you will find this. So significant, usually students study about mitosis, study about meiosis, but they forget to read about the significance of mitosis and significance of meiosis, which is clearly given in a separate paragraph in NCERT. So go through that. Question is asked from there. Then a bivalent consists of, first of all, let's understand what is a bivalent, when it is formed, right? So pairing of two homologous chromosomes will cause formation of bivalent. So bivalent, let's say this is one chromosome, with two sister chromatid, another chromosome with sister chromatid, right? So when the pairing take place, so pairing, now here if I am drawing it like this, then it seems a little difficult to figure out. Now I will, if I draw it like this, let's say this is one chromosome with two chromatid, then here again one chromosome with two chromatid, and now they are paired. Now they are paired with each other. Now they are paired. So here these two are non-sister chromatid and these two are sister chromatid. So now here this will be called as a bivalent. So how many chromosomes are there? A bivalent has two chromosomes. How many chromatids are there? 1, 2, 3, 4, two sister each. So 4 chromatids are there. How many centromere are there? 1 centromere, 2 centromere, right? So 4 chromatids and 2 centromere. See, 2 chromatid, no, 2 chromatid, no, 4 chromatid and 2 centromere. 4 chromatid and 4 centromere is wrong. Next, very interesting question. Little bit of calculation is there, but such questions are very important. Now, if we see here, bacteria divide after every 35 minutes, and I am going to tell you the trick to solve all such questions, right? So here, basically, what is the doubling time if I ask? Read the question carefully and tell me what is the doubling time. What is the doubling time? So just by reading, bacteria divide every 35 minutes, means every 35 minutes 1 become 2, means doubling time is 35 minutes. Now what is the total time? Total time is 175 minutes. Doubling time is 35 minutes. Total time is 175. So number of doubling will be, right? So number of doubling in 35 minutes, 1 doubling. So in 175, so number of doubling, can we say n is equals to 175 by 35, right? So this is the number of doubling. So it will be 5, isn't it? When we have 5, then number of cell after 175 minutes, after the final time, it will be the number of initial cell, means initial number of cell, whatever it is, 1. So here what is the initial number of cell? 10 to the power 5. So it is initial number of cell, into 2 to the power n. N we have seen 5. So here initial number of cell is 10 to the power 5 into 2 to the power 5. So can we call it 32 into 10 to the power 5. This is the total number of cell. So initial number of cell is 10 to the power 5. Now here if I would have said 1 cell, then 1 into 2 to the power n, that is 32, right? So remember, total time divided by doubling time will give you total number of doubling, and then make it 2 to the power n and multiply with the initial number, you will get the final number of cell. You will get the final number of cell. Clear? Now let's move ahead to the next part here. Cell cycle. Cell that do not divide further exit G1 phase to enter an inactive stage called. So we all know this part, that there is a M phase, then there is a G1 phase, S phase and G2 phase. So although here just if I change a little bit and I make this M a little long. So this is G1 phase, S phase, G2 phase, right? And here M phase also have prophase, metaphase, anaphase, telophase, cytokinesis, etc. So now in the G1 phase, this is M phase. Now this is, I am drawing intentionally so that you can revise. From this diagram also questions are asked sometime. So G1 phase only cells exit out and get a step into the G0 phase. So this is what? Cell do not divide further, exit G1 phase to enter into an inactive stage, and this inactive stage is called G0 stage. This is also called quiescent phase. Quiescent phase it is called. Next one, select the odd one out with respect to mitosis. Odd one out with respect to mitosis. It helps the organism in both sexual and asexual reproduction. Odd one with respect to mitosis. So mitosis help organism in both sexual and asexual reproduction. It is called equational division, right? It is called equational division. It take place only in diploid cell of plant. And lastly, it help in cell repair. Now, see, it helps in cell repair is correct. It is called equational division is correct. It helps in organism in both sexual and asexual reproduction. Yes, so mitosis not only help in asexual. Mitosis do take during sexual reproduction also, although meiosis also is there. So this statement is also correct. Now it take place only in diploid cells of plant. Now it's not correct. It may take place in haploid cells also, right? Although in higher plant mostly it take place in diploid cell, but it can also take place in haploid cell. So this statement is wrong. Rest of them are correct, right? Then plant cell division. In plant cell division, the formation of new cell wall begin with the formation of simple precursor called. Now here remember, I will tell you the flow chart through which the cell wall formation take place. See, first of all, there is a phragmoplast. Phragmoplast is formed, and this phragmoplast is formed with the help of Golgi apparatus, right? This phragmoplast forms the cell plate. This cell plate develop into middle lamella, and then this middle lamella develop into cell wall, which will have primary wall and which will develop secondary wall. So this is the flow chart for the formation of cell wall. So here if I say cell plate, so the cell plate that represent the middle lamella, cell plate itself develop into middle lamella, right? So cell plate disappear or it gradually get converted into middle lamella. Then middle lamella remain as it is, additional layer of primary wall is formed, then additional layer of secondary wall is formed. So the cell plate that represent the middle lamella, the cell plate that represent the primary wall, the cleavage furrow that represent the middle lamella. During cell wall formation, furrow is not formed. It is the cell plate which is formed. So again wrong. The contractile ring that represent the primary cell wall. This is also wrong. So first, correct? Then, how many of the given statement are correct? All bacteria are autotrophic, right? All bacteria are autotrophic. Wrong. Bacteria can only survive on dead organic matter. Again, this is wrong. All bacterial cells are surrounded by a thick peptido. This is also wrong. Mycoplasma, without cell wall, it is a type of bacteria. So all three are wrong. Methanogens are archaebacteria. Methanogens are archaebacteria, correct? Blue-green algae are unicellular algae. See, they can be unicellular, they can be filamentous or colonial. All three can be possible. Mycoplasma are true bacteria. Now, here, mycoplasma are true bacteria. What are true bacteria? True bacteria means eubacteria. So, they come under eubacteria. So, this is also correct. So, A and C is correct, B is not correct. How many of the following statement regarding the fungi is true? Asexual reproduction is commonly by formation of spore. This is from the fungi, biological classification we are moving in. Their bodies consist of hyphae which form mycelium, again correct. They secrete digestive enzyme onto organic matter and then absorb the product of the digestion, correct. Fungi can break down almost any carbon containing product. Yes, almost any. There are some carbon which they can't. So almost, so that is also right. Fungi do not enter symbiotic relationship. This is totally wrong, right? They form symbiotic relation, lichens and mycorrhiza. Now, cymose type of inflorescence. Now here we are talking about the morphology. So here what we see, inflorescence is of two types, cymose and racemose. Cymose and racemose. So in case of cymose, what we see, that the branch will end with a flower, right? Every branch will end with a flower, means flower has a terminal position, right? And new flower always at the bottom. New flower at the bottom. This is about the cymose. Now, so here if we see, main axis continue to grow. Main axis terminate in flower. Main axis do not continue to grow. Wrong. Main axis continue to grow. Growth is limited. Yes, growth is limited. And flower are born in the basipetal succession. This is correct. Acropetal is for the racemose. Remember, acropetal. And here it is basipetal. Cymose is basipetal. Then here it will have limited axis, right? And here continue to grow. Here we say axis continue to grow. Then flower position, here it is terminal, here it is lateral. So keep it in mind. These are the main difference between cymose and racemose. Now regarding taproot, we know taproot are of two type. Root are of two type, taproot and fibrous root. There is adventitious root also. So here taproot, in taproot there is one main root which is called primary root. Then it will have a secondary root. And then it will have a tertiary root. Primary, secondary and tertiary root. Secondary and tertiary are called lateral root. They are called lateral root. So here the primary root and its branch constitute the taproot system. Correct? taproot of sweet potato. Now, sweet potato is a taproot? No. Read NCERT. It is adventitious root. Sweet potato is adventitious. So, statement 2 is wrong. Statement 1 is correct. Then, maxillary estivation. So, last few question is left from morphology. So, here in maxillary estivation. So, maxillary estivation is what? When the petals are arranged in this manner. And this we call as 1 plus 2 plus 2 in this way. Here this petal is called standard. Standard. This side one is called wing and this one is called keel. So here in symbol 1 is the standard. This 2 is the wing and this 2 is the keel, right? So on that basis, the maxillary estivation, the standard overlap the wings. Yes, standard overlap the wing. The standard overlap the keel. No, this is wrong. The standard is overlapped by the keel. Keel is here, standard is here. Again wrong. The keel overlap the wing. The wing overlap the keel, right? So these are 3 statements. So first one is correct. Then last question. China rose is not characterized by China rose, right? So if we say China rose has superior ovary. Ovary has a highest position. China rose it is monoadelphous stamen. All the bundles are together in one, all the stamens. So yes, it is monoadelphous. Estivation is valvate. China rose, estivation is valvate, right? So valvate estivation, this is correct, right? Valvate estivation is there in China rose, or is it twisted estivation in China rose, right? So China rose has twisted estivation, right? So China rose has twisted estivation. So China rose is characterized by twisted estivation. So that part is wrong, right? So with this, the part of morphology is covered. So I hope that you all have got some idea what and how to solve, right? These all questions are very much relevant to score as well as NEET. Go back to the topic and revise these topics once again. And you should be noting it down that the score exam is on these dates. Links are there in description. You can just note it down, go through it, download the syllabus. Not registered, quickly register and be the part of the largest Olympiad, right? It is kind of a largest exam in which one of the brightest mind from all corners of India will be participating, and it is one best opportunity to test your knowledge. So just continue to do good. Thank you all of you.